Okay, so folks, this is Open Lab, October 25th. This is the third little movie, and we just want to state that we're trying to do some review topics, but it's not exhaustive. So in the previous movies, we did an antibiotic review, then we did glycolysis, fermentation, aerobic respiration. And so this movie is going to be DNA replication, which I know is really hard in lecture. It's a dynamic process, and we are trying to draw it on the board. And I think a lot of people didn't feel that great about it. So we just brainstormed and said, why don't we start with just putting a list of the proteins and enzymes involved in DNA replication. And if you know the functions of each of the proteins and enzymes, that should be really big, big understanding. Okay, so we're doing DNA replication in E. coli. So remember, you guys, that there is an origin of replication in the circular chromosome of E. coli, and it's at the origin of replication that initially we'll see that replication bubble form, and there'll be two replication forks where all the proteins and enzymes are going to be at work, using the two old parent strands to make two new daughter strands. So that's what we'll do, is we'll be looking at all the proteins and enzymes involved. So first of all, folks, remember the chromosome was supercoiled, so what was the first enzyme that we needed to relax the supercoiling?
Bacterial gyrase.
Good, bacterial gyrase. And that's going to relieve the chromosome supercoils. Okay, all right, so that's good. And then we need to take our old parental double-stranded DNA, the chromosomal DNA, and we need to make two single-strand templates. So we have to break the hydrogen bonds. We have to unzip the double-stranded DNA to make two single-strand templates. And who's going to do that?
Helicase.
Awesome, helicase. Good. Oops, helicase. Helicase. We can say, is it too unscientific, you guys, to say unzips the double-stranded DNA of the parent chromosome? And what will we make, you guys, when we unzip the double-stranded
Two
DNA?
single-stranded.
To make two single-strand DNA. What's the term? What's a fancy term for a guide?
Templates.
Yeah, exactly. You'll see that a lot. Templates. Good. Okay. So now we have our single-strand DNA templates. So what is a danger, you guys, in having single-strand DNA?
It can tangle and get
right?
destroyed, hydrolyzed.
You'd be hydrolyzed, right? And it can come back together again.
Yeah.
So who will protect the single-strand DNA templates?
Single-strand binding protein.
Awesome. So this SSBPs, single-strand binding proteins. Single-strand binding proteins. They bind to the single-strand DNA and protect it. Protect it from reannealing, coming back together again, protect it from tangling, protect it from being hydrolyzed by the bacterial enzymes that might recognize it. Oh, it looks like bacterial viral DNA. Good. And then, folks, we said that DNA polymerases, this is actually linked, because DNA polymerases proofread. They can't just land on single-strand DNA and start synthesizing complementary DNA. They have to have a little primer there so that they can check to make sure the base pairing is correct. So what was the name of the special RNA polymerase that will make a short little RNA primer?
Primase.
Primase, good. And we want to remember it's a special type of RNA polymerase. And because RNA polymerases don't proofread, they can just land on DNA and start making complementary RNA. And its job is, which makes a short RNA primer for DNA polymerase, because DNA polymerase needs a short little primer to get started. Okay, and then once primase has made the short little RNA primer, who's the workhorse in E. coli that does most of the DNA synthesis?
The DNA polymerase 3.
Good. Yeah. So DNA polymerase 3 is our workhorse, going to do most of the DNA synthesis. So it's gonna make complementary DNA using the single strand DNA as a template. Okay. And on the lagging strand, once DNA polymerase 3 starts synthesizing, it just keeps going until it goes around to like the opposite side of the chromosome to the termination of replication. Okay. So that was easy, the leading strand, right, continuous DNA synthesis. But I think where the headaches, what was the headaches, you guys, was that lagging strand, right?
Mm-hmm.
So remember, we'll just do that right now, you guys. So the leading strand is made using continuous DNA synthesis. So that wasn't too bad, but it was a lagging strand, right? It was a headache. And so the strategy to synthesize a lagging strand, you remember what the strategy was?
To discontinue.
Yeah. So the strategy the cell uses discontinuous DNA synthesis, where DNA polymerase starts and then it has to stop, and then it starts and it stops. And it's a little bit more complicated. Okay. So the lagging strand using discontinuous DNA synthesis, I mean, it's going to, all of these enzymes and proteins are required, right? But we needed some extra ones. And one of the extra ones was a DNA polymerase that can remove the RNA primers and replace them with DNA. So remember, DNA polymerase 3 can't do that. So who helps remove the RNA primers and replace them with DNA?
DNA polymerase 1.
Yeah, exactly. DNA polymerase 1. Yeah. So DNA polymerase 1, it removes the RNA primer and replaces with DNA. And the problem is on that lagging strand, we have all these DNA fragments, though, called Okazaki fragments. So the problem is DNA polymerase 1 can't form a covalent bond between the DNA it's synthesizing and then it runs into an Okazaki fragment in front of it. It can't form a covalent bond there. So what's the enzyme that covalently links Okazaki fragments together?
Ligase.
Yeah. So we'll put a DNA ligase covalently links the Okazaki DNA fragments. And then once DNA polymerase 1 and ligase are finished, you can't tell that the lagging strand was at one time, alternating DNA RNA or little DNA fragments. It looks just like the leading strand. Yeah. So you guys, do you think it would actually make it? Maybe my mistake was in trying to cartoon it, because I think maybe the cartooning made it harder to understand. Do you think it's okay to leave it like this?
It's easier because it's step by step.
Exactly. Yeah.
As long, I guess, as it's not going to be the picture, like for us to recognize the picture.
There probably won't be a picture of the DNA replication fork. Remember, you guys, how we did it in lecture? We had our problems and solution table. Yeah. I mean, that would probably be really helpful. And I think I said in lecture, that's often, at least one way I ask you about this. Mm-hmm. Here's the problem. What was the solution? Okay? So you probably won't be labeling a picture. For me, it's just because I'm so visual, I have to draw everything to see if I understand it. And that's why, unfortunately, I kind of impose that on you guys, and I'm a lousy drawer. So sometimes that makes it worse. It doesn't help because my drawings aren't that good.
Okay. Well, I think...
Would the question be like in short answer form, or do you think you're going to have like a multiple choice type?
It could be either. It could be either, yeah. And I know that's so frustrating, you guys, but if you're prepared for both, then you're really prepared. Yeah.
So for the supercholine, the antibiotic that targets that supercholine was a ciprofloxacin.
That's right. The quinolone, the fluoroquinolone, ciprofloxacin is going to knock out the bacterial gyrase. So gyrase can't relieve supercolon, you can't get DNA replication, you can't get binary fission, and furthermore, you can't get transcription because you have to be able to separate the two strands of DNA for transcription, and if the chromosome is supercoiled, you can't transcribe the DNA either.
Right.
Yeah. Yeah. So you guys, we've got 10 minutes left. Should we do the transcribe the DNA and then translate the DNA? Does that sound all right, you guys? Okay. And remember, folks, there'll be a genetic code table on the exam, right? So you don't have to memorize any of that. And do remember, you guys, there might be other questions in the multiple choice section where your genetic code table might help you. So just remember, it's at the back of the exam and it's there to help you throughout the exam. Okay, so you guys, let's do, good. Let's do a practice transcription and translation. And remember, you guys, we said in lectures, this definitely will be a short answer question. The genetic code table will be attached to your exam. Okay, folks, and I'll set up the DNA sequence correctly. I'm not trying to trick you by having the DNA in the wrong orientation. So you guys, so we're gonna, I'll give you a DNA sequence and I'll set it up correctly. Okay, so I've oriented the DNA strand correctly. So now, sometimes people get confused with the direction. So folks, if I say transcribe the DNA, what are we going to make? The DNA is our template. What are we going to make?
mRNA.
Yeah, in this case, mRNA, because we're eventually heading for protein. Yeah. So you guys, and again, I set it up correctly. So this is going to be a 5' of mRNA. So you guys, what will be the mRNA sequence?
So A, U, G, C, G, U, C, 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 U, A, G.
Good. Okay. Perfect. Okay. And then, folks, I'll say translate your transcription product.
So
translation is using mRNA as a template to make protein, right?
Yeah.
So, and again, folks, remember you have to have your genetic code table. And folks, I don't have it. Do you have one there?
I have it,
Awesome.
yeah.
Can I borrow it? Crystal, thank you so much. Okay. Perfect. Thank you so much. So remember, you guys, we're going to have a genetic code table on your exam. So you don't have to memorize this, but you have to know how to use it. So, folks, what we're going to do in translating our mRNA into protein, we're going to do it one codon at a time, one triplet at a time. So AUG, if I look at my genetic code table, AUG, it says it's going to be translated into methionine. Or, folks, if I tell you this is transcription and translation in E. coli, and we presume that's the start codon, if you tell me formal methionine, I'll give you a bonus point. So don't even worry about it, but just remember in bacteria, the first AUG, the start codon, is translated into formal methionine. But again, don't worry about it. It's like half a bonus point, something like that. Good. And then, folks, CGU, so CGU is arginine. Okay. And then CC is proline.
UAG stop.
Good, excellent, you guys. So UAG, you see it's a stop codon, right?
So you don't
And
even
so
write stop,
you don't
right?
even have
Okay.
to write it down, or if you want to. I mean, it's fine. You know, some people like to put stop. You know, it's just like point, right?
But that's not going to have the covalent
No,
bonds.
yeah,
Okay.
right. So you can just leave it blank or just put stop, okay? And then the flip one, and this would be bonus, you guys, if I give you the tRNA anticodon. So this is what I have to do myself because I get mixed up.
Okay.
If this is and again, folks, this is just a bonus question. if I give you a tRNA anticodon, so if given a tRNA anticodon, which amino acid would the tRNA carry? And the key to this, folks, is I think, for myself, I have to draw the cartoon. So let's say the anticodon that I'll give you is going to be, and I'm going to cheat here, you guys, I'm going to go GCA, right? So again, folks, even making the key, I have to draw the cartoon, because otherwise I'll get myself confused. So my anticodon is GCA. So the key is you have to draw the mRNA codon to which that tRNA anticodon will bind. So what would that be?
So it'd be
CGU, right? And then the key is, folks, then you look for this mRNA codon on the genetic code table. So can you help me out?
Yeah.
CGU.
It's argon.
Yeah, and see, I cheated you guys because I was.
Oh, yeah.
Okay. Anyway, but it's good. So then this tRNA is going to carry arginine. And again, the mistake that folks make are the instinct, and especially when you're exhausted and tired on the exam, is the mistake people make is they look for the anticodon on the genetic code table, and you're right, you have to do that little step, find out the mRNA codon to which the tRNA binds, and then find on the genetic code table the amino acid that this mRNA codon will be translated into. Okay. So, folks, again, this was not exhaustive. Other things that, if we had more time, we would, in metabolism, and again, I was kind of focusing on metabolism because I was just worried it was so long ago, but in metabolism, we would have mentioned anaerobic respiration, where there's another electron transport chain, but the terminal electron acceptor is not oxygen. It's some other inorganic molecule like nitrate or sulfates. As a follow-up on that, remember you guys, I gave you that nitrogen cycle handout, and there were some applications down at the bottom. I said, make sure you read these because I love to ask this on the lecture exam. So we wanted to talk about how farmers like nitrogen fixation and nitrification, because those are forms of nitrogen plants can use to grow, but farmers don't like denitrification. That's when your soil becomes waterlogged, it becomes anaerobic, and the bacteria switch to anaerobic respiration using nitrates as the terminal electron acceptor, and the nitrates can get converted back to molecular nitrogen, which is not usable. The plants can't use it, so that's called denitrification. So farmers don't like denitrification, but if you're working at the sewage treatment plant, you like denitrification. You want to get rid of usable nitrogen in the treated sewage before you dump it into the rivers. Because if there's excess usable nitrogen in treated sewage, it causes those harmful algal blooms or cyanobacteria. And another place that we would want denitrification is that if farmers are using artificial fertilizers containing nitrates and they're using too much. So if the farmer irrigates or there's a rainstorm, nitrates wash into our waterways and then end up in the coastal waters. That too will cause harmful algal blooms. So they're using bioreactors to run the irrigation water after it comes off the fields. The bioreactors are used, so the bacteria in the bioreactors will do denitrification to convert the usable nitrates into gaseous molecular nitrogen, and then you let the water flow into the waterways, and you won't get the harmful algal blooms that way. So it kind of depends on your point of view, but the that nitrogen cycle handout, there's probably going to be at least 3 or 4 questions on the nitrogen cycle and applications.
Okay.
Yeah. And then, folks, we didn't talk about microbial growth at all. I mean, there's so much we haven't talked about. But again, my thought was metabolism. Like it seemed so long ago. My thought was just kind of reviewing it. And also, you just want to know the difference between oxygenic photosynthesis and anoxygenic photosynthesis. Okay. So, any final questions, you guys? Sure.
How do you remember like the names? 'Cause I'm going over the
Oh, yeah, no,
the
Alex, you
amino
don't have
acids
to,
on the
yeah,
top.
you don't have to memorize the names of the amino acids. I'll give you a table that has the full name. Or if I gave you a table and all it had was the abbreviations, then all you need to do is use the abbreviations, right? It's just whatever is on the table I give you. Yeah, okay? Okay. So we'll end this, and just wishing everybody the best of luck on the lecture exam to try to get some sleep. That's so important. Try to eat before you come.
That was the
Bring
hard
number
thing
2 pencils.
about the lab exam. We went from lecture to lab with like no time to eat. Or
I know, it's so true, isn't it? I know that, and you know, as instructors, we've said those 10 minutes between classes, just it's not enough for anybody. Oops, oh sugar.
I know since we're out of time,